This came out of this study came out of another epigenome-wide association study, and in this study, the difference to our previous and other studies was rather than looking at individual sites within the genome, we tried to clump them together to try and identify regions consisting of multiple neighbouring methylation sites. And when we did this, we found this one block which showed increased DNA methylation with Alzheimer's disease pathology. And interestingly, it spanned 48 kb of the genome, and it contained 208 of the methylation sites we looked at. So it seems to be quite a substantial change, and it seems to be quite specific to certain areas of the brain. So we identified it in the prefrontal cortex, but it doesn't seem to be altered at all in areas of the brain such as the entorhinal cortex, which are typically regions that actually you would start to look at in terms of methylation profiling. So that's quite an interesting thing we're also following up as well. In terms of following up on that, we're doing targeted sequencing of that gene, and actually ANK1 as well, so to look at every single methylation site in the genes. We're also looking at gene expression of it as well, and then also doing some chromatin immunoprecipitation, PCR, to actually look at levels of histone modifications within the gene too, to try and get a complete picture of what's happening with this gene in terms of its regulation in Alzheimer's disease.